Divide and conquer. It is not just a strategy used by politicians to take over the country, but also a strategy used by teeny tiny microbes such as bacteria to take over the world. So much so that these bacteria make up to 50% of human body cells. One of the things you must have heard about these bacteria is that they are haploids. It is mentioned in every book. Well, before judging someone's ploidy, let's understand what it is. Ploidy is nothing but the number of genome copies an organism has. If it has one copy, it is an haploid, two copies, a diploid, and polyploid if it has more than that. The haploid organisms, such as bacteria, disagree that they are haploid. To understand why, let's look at the role model, the E. coli. E. coli has a special problem that it has to divide in 20 minutes with a genome of 4.6 MB, and before dividing, it has to duplicate its genome. Well, why is that a problem? To understand that, let's look at this. This genome of E. coli is replicated with the help of enzyme DNA polymerase III, which has a rate of 1000 base pairs per second and processivity of 50,000 base pairs. If you consider the circular genome of E. coli with its one origin of replication and its bidirectional replication mode, each replication fork has to divide 2.3 megabase per DNA. And if we consider the rate of replication, that is 1000 base pairs per second, it will approximately take 2300 seconds to divide the whole genome, which comes around 38 minutes. Now that is the problem. It has a doubling time of 20 minutes experimentally seen, but it requires 40 minutes to replicate its genome. In 20 minutes, it cannot replicate its whole genome. It can only do it halfway. Well, in the log phase in which bacteria divides in 20 minutes, it can sense that there are many nutrients which are available and bacteria can grow faster. And after understanding that, what bacteria does is it starts the replication on again on both the full genome and the half divided genome. So by the time bacteria divides, it already has half divided genome and it only needs 20 minutes to divide another half. So every time bacteria grows in the jar with 20 minutes of doubling time, it always has 1.5 times the genome and not the one copy of genome which has been taught in books and schools. So they are not actually haploids, they are 1.5 haploids. Let's stop assuming that E. coli is haploid, and maybe in the next protein prep it will not irritate you with the OD. Thanks for watching.